Thank you everyone for joining. This is our second webinar for 2021. As you all know, the topic for today's webinar is using single-cell data to deconstruct a human autoimmune disease, rheumatoid arthritis. So hopefully you'll have questions. If not, please prepare for the Q&A session after Professor Brenner's presentation. I want to introduce to my co-host, who is Corinne Solier, who is actually Skylight's Chief Operating Officer. Corinne just joined Skylight from Roche and brings a solid experience in translational drug development and biomarkers. Now I give the mic to her so she can introduce today's speaker.
Thank you, Kaloyan. Can you hear me fine now? Very good, thank you. So good afternoon, everyone. Thank you for joining us today. As Kaloyan mentioned, I'm Skylight's Chief Operations Officer. Skylight was founded in 2017 as a spin-off of the ETH in Zurich and is based in Basel in the Swiss Innovation Park. Skylight's proprietary platform is called SkyVision. It is a neural network-based algorithm that gains clinically relevant insights from high parametric data like single-cell RNA sequencing and from medical data as well. We are collaborating with companies developing cell and gene therapies, immunology and oncology therapies, which we can complement and support. And we do so by leveraging our best-in-class artificial intelligence SkyVision platform with single-cell data analytics to increase the probability of success of their molecules and by reducing the development cycle times. Today, we are extremely honored to have Professor Brenner for this webinar. Professor Brenner is an advisor to the board of directors of Skylight. He is the director of the Brigham and Women's Hospital's Human Immunology Center, which creates and performs single-cell transcriptional analysis and functional investigations in order to decipher human autoimmune illnesses, stratify patient groups, and find prospective therapeutic target cells and pathways. He is a member of the National Academy of Sciences in the United States and has received numerous prestigious awards for his groundbreaking research. Professor Brenner's research focuses on T-cell biology, antigen presentation, microbial pathogenesis, and autoimmunity. In the course of his research, he discovered the integrin molecule AEB7, also called CD103, and its role in leukocyte homing, as well as a role for the CD1 antigen presentation system in the activation of T cells and NKT cells. Most recently, his team has identified distinct fibroblast subpopulations that are pathologically expanded in disease, as well as a new T helper cell population that are involved in antibody production in autoantibody-mediated disorders. From a logistical point of view, we will be listening now to Professor Brenner's seminar, and this will be followed by a Q&A session. Please make sure you are muted during the whole talk, but you can already start asking your questions in the Q&A chat box that you can open from the bottom right side of your screen. I look forward to the discussion that we will have after your talk. Professor Brenner, I give you the stage now.
Okay, can you see my screen?
Yes.
Yes, okay. Great. Well, it's a pleasure to join you to talk about using single-cell data to deconstruct human autoimmune diseases, and I'll use rheumatoid arthritis as the prototypic example. First, to introduce RA, as shown here on the left, this is the hand of a patient with severe RA. You can see the massive swelling and hyperplasia in the joints, the wrist, the knuckles, and the destruction to the cartilage and bone underneath that occurs in severe RA. Now, the localization of the site of this process is in what's called the synovium, and that's pictured schematically here. It's this little thin membrane shown around the edges of the joint that completely encapsulates the joint. And in the normal synovium here on the upper right, it's a very bland looking thin structure. It has a lining layer of just a few cells thick, and then the sublining layers, loose connective tissue, adipocytes, and blood vessels. Its normal function is to provide lubrication, lubricin, hyaluronan, and nutrients to the synovial fluid. On the other hand, as shown down here in the lower right, in rheumatoid arthritis, this tissue becomes massively inflamed, infiltrated in the sublining with leukocytes, and the fibroblasts, which make up the lining and sublining, undergo massive hyperplasia. And then this is the tissue that then destroys the cartilage and bone and causes pain and disfigurement. Well, I'd like to start by framing the way we think about the inflammation in rheumatoid arthritis by talking about T helper cell subsets. So most people have considered CD4 T helper cells to be the major drivers of rheumatoid arthritis. And there's a number of different T helper cell subsets: Th1, T helper 1, T helper 2, T helper 17s, etc., are well known in immunological studies. And most people have considered RA to be a Th1 disease, meaning it's driven by CD4 T helper cells that make interferon gamma, or it's been considered to be a Th17 disease, where T cells that make IL-17 related cytokines drive the disease. Well, we took a different approach, essentially a discovery-based, unbiased approach using single-cell technologies to simply ask, what do we find when we look at RA in an unbiased way? So we started using CyTOF as a single-cell cytometric technology, and shown here is a t-SNE plot of the CD4 cells circling a group of cells that have very high levels of expression of CD1. Now, CD4 cells that express high levels of PD1 are well known in the immune system, and they're depicted here from this cytometric stain of tonsil, staining for CXCR5 on the x-axis and PD1 on the y-axis. And these cells that are PD1 high and express this chemokine receptor CXCR5 are typical for T follicular helper cells, TF8 cells, that drive B-cell differentiation in follicles. But that's not what we found in the synovium. Over here to the left, what we found in the synovium were PD1 high cells, but not the ones that express CXCR5. They lacked CXCR5. And if you look at their quantitation, they accounted for 25 to 30% of the CD4 T cells in the synovial fluid or synovial tissue. And when we asked what they made as effector molecules, they produced high levels of the cytokine IL-21, which drives B-cell differentiation, and CXCL13, which attracts B cells and helps organize follicles. When we tested these cells to ask if they could drive B-cell differentiation to plasma cells, we found that in fact they could. And so since these cells are in the periphery, in the synovium, not in lymph nodes in the follicles, we named them TP8 cells, or T peripheral helper cells. And this is schematized here. Previously, we knew that T cells are important for driving B-cell differentiation to generate antibody production. What we knew before is that T follicular helper cells, located in the follicles of lymph nodes, would then interact with B cells, produce cytokines and chemokines like IL-21 and CXCL13. These cells both express CXCR5, and so they come together in germinal centers in the main germinal center antibody reaction. What we found in the synovium in RA is that cells coming from the periphery that express peripheral homing chemokine receptors like CCR2 localize in the synovium. But once they get there, these cells function a lot like T peripheral helper cells, like T follicular helper cells, producing the same chemokines and cytokines that drive B-cell differentiation, but this is occurring in an extrafollicular location. And this is a fluorescent stain of synovium staining T cells in red and B cells in green. And basically, you don't see very many organized follicles. You see aggregates of T cells and B cells. And we think this is what a lot of the T cells in rheumatoid arthritis are doing. They're driving B cells to produce antibodies and autoantibodies. Now, we've taken this same kind of discovery-based approach at the consortium level. This is a consortium called the Accelerating Medicines Partnership, or AMP, RASLE, which is designed to do a single-cell disease deconstruction by taking biopsies of synovium in RA and kidney in lupus nephritis and subjecting them to single-cell analyses in an unbiased way. So synovial biopsies are collected from many, many sites. Histology and blood analyses are carried out. And the main pipeline I'm going to talk about today is the single-cell pipeline, where the synovial structures are disaggregated and then stained with antibodies that have barcodes, so we can get a protein library of cell surface proteins, and also a single cell RNA, mRNA library using droplet-based capture using the 10x genomics platform. So we get two single cell libraries on the same cells to analyze. At the same time, we take an aliquot of cells, subject them to flow cytometric sorting, and then get bulk RNA-seq for different cell types, fibroblasts, monocytes, T cells, and so on. And then in this very extensive pipeline, we also get single cell attack-seq and immune repertoire. Today, I'll talk mainly about this main pipeline, and I'll start with the flow cytometry and bulk RNA-seq on these synovial cells from rheumatoid arthritis patients. So when we look at the genes that are most upregulated in patients with inflammatory or leukocyte-rich RA compared to osteoarthritis, these are the genes we find that are most upregulated in RA synovium. And interestingly, in the sorted T cells, and interestingly, the dominantly overexpressed genes are CXCL13. That's the chemokine I described coming from the TP8 cells. And here's PDCD1, the gene for PD1. And so this signature of the genes at the asterisks really are hallmarks of the TP8 cells that I just showed you we had identified independently from the CyTOF analysis. And if you look at the single cell RNA sequencing data from the RA synovium, from the first phase of the AMP consortium analysis, and you ask which population of cells is making the CXCL13, you can see it's all coming from the population of TP8 cells. Now, in addition, we saw high levels of interferon gamma. As we mentioned before, RA has been considered a Th1 disease with the interferon gamma being prominent. But interestingly, when we look at the single cell data shown here on the lower right, it's not the CD4 T cells, which are here, that are making the interferon gamma. It's mostly coming from the CD8 positive T cells, really quite a surprise. Now, if we look more closely at the deep clustering of the T cells, as shown here in this UMAP space, we can see that we identified 24 different T cell clusters from about 94,000 T cells in this AMP consortium. And I won't go through all of them, but just to give you kind of a broad brush of what cells are there, most of the CD4 T cell clusters are here to the left. The CD8 T cell clusters are here to the right. If we look more closely within the CD4 cells, there are two clusters of TP8 cells. One of them has TP8 cells alone, and the other is a cluster that mixes TPH and TFH, these two helper cells for B cell differentiation. There are two populations of Tregs, one that expresses CD25 and one that lacks it. There are some naive T cells, several types of memory CD4 T cells. On the CD8 side, we expected to find mostly CTL, cytotoxic lymphocytes, that express granzyme B, perforin, granulysin. And of course, we did identify them in this cluster 15, but it was actually a relatively small proportion of the CD8 T cells. A larger proportion of the CD8 T cells included several clusters that expressed a different granzyme, granzyme K, sometimes also with granzyme B, but especially marked by granzyme K. Other populations included gamma delta T cells, which express granzyme K, and naive T cells. Well, we were struck by this granzyme expression difference because we really expect mostly to find granzyme B, but instead we found more granzyme K. And if we look at the expression pattern in this same UMAP, now shown here to the right, you can see granzyme B expressing CD8 T cells here, and the much larger, more prominent CD8 population expressing granzyme K, indicating that it's really the granzyme K population which is the major tissue CD8 T cell in the synovium. And we don't know much about this population. It hadn't really been identified as the predominant CD8 population before. And so Helena Johnson and Fan Zhang in my lab and in the lab of Shomura Chaudhry looked at this a little bit further. And this just confirms a quantitation by flow cytometry that CD8 T cells make up 35 to 45% of the T cells in the synovium. So it's not quite as many CD8 cells as CD4 cells, but it's almost half. Importantly, however, when we ask which T cells are making the most interferon gamma, it's really the CD8 T cells that have a higher percentage of them producing interferon gamma than CD4 cells, quite surprising. And this is just showing in permeabilized staining that indeed a majority of the CD8s in RA synovial fluid and tissue are indeed granzyme K. Now we then generated a granzyme K, granzyme B score and could apply it to the single cell RNA sequencing data from a number of diseases. And as you would expect, and as I just shown you, when we apply this score to rheumatoid arthritis or osteoarthritis, these orange bars represent the granzyme K score and the blue bars the granzyme B score. And as you can see, granzyme K cells predominate in RA, as we found using the other methods. Interestingly, when we look at the gut in ulcerative colitis and Crohn's disease, again, it's the granzyme K T cell score that dominates over the granzyme B score. And this applies across other diseases I won't show you. So these granzyme K CD8 T cells really are the major tissue CD8 T cell form. Now let's go from T cells to B cells. This is the UMAP of the B cell clusters that we're seeing. And if you notice on the left here are plasmablasts and plasma cells. These are the plasmablasts and several types of plasma cells. And on the right, you can see the B cell clusters. There are several types of memory B cells, naive B cells. I'd like to focus on two populations of B cells. The GC B cells, which are the germinal center B cells. So these would be the ones in follicles, like in lymph nodes, but in this case, they're in the synovium. This is referred to as tertiary lymphoid structures. But there's a small number of those and a much larger number of ABCs. This stands for age-associated B cells. These are CD11C high, Tbet high, make interferon gamma. These B cells are associated with being pre-plasmablasts, and so they may give rise to these plasmablasts. And they've also been linked by their phenotype to be similar to DN2 cells, which have been proposed to undergo differentiation in an extrafollicular pathway. So we're seeing B cells that may be involved in extrafollicular B cell differentiation to plasma cells. Now, very importantly, the single cell analysis of the synovium led us to realize that you could segregate patients into different phenotypes based on the abundance of different types of single cells in their synovium. And we've called these CTAPs, for cell type abundance phenotype. And we think this is a new way to stratify patients. And so these single letters here refer to the cell type that dominates in the CTAP, in the subset of RA patients. So some of them, for example, have a high population of T cells. But in this case, the patients have a composition of their synovial tissue that's mostly T cells and fibroblasts. Compare this to this population, this group of patients, who mostly have T cells and B cells, which is different from this group of patients that mostly have T cells and macrophages. And actually, when we look at principal components analysis, you can see that most of the variance in the first principal components has to do with the populations of patients who have mostly T cell rich synovial infiltrates. Now, on the other hand, here's a group of patients that don't have so many T cells, but mainly have a myeloid cell rich infiltrate. And the second principal component, the lower part compared to the upper part, are the CTAPs that mostly have myeloid cells. You can see here is a group of patients whose cell type abundance phenotype is to be mostly composed of fibroblasts, this population, and this population mostly have endothelial cells, fibroblasts, and myeloid cells. Both of these have very few lymphocytes compared to these, which are lymphocyte rich. So we think this is a novel way that we can really begin to stratify RA patients. And an important way we can use this is by applying a new algorithm called CNA. CNA stands for covariating neighborhood analysis. And this is an analysis where we identify groups of small numbers of cells in small regions in transcriptional space that we term neighborhoods. And then we can determine if these neighborhoods of cells covary in abundance with some attribute. And the attribute in this case would be these unique stratifying CTAPs that separate the RA patients. So for example, if we look at the patients whose synovial infiltrate is mainly composed of T cells and B cells, the TB CTAP, we can ask which particular type of T cells is expanded in that tissue type of rheumatoid arthritis. And what we see here in red is it's mostly those TPH cells, those T peripheral helper cells, or the combination of TPH and T follicular helper cells I talked about at the beginning of the presentation. Interesting look in comparison, if you look at the histologic type of infiltration of mostly T cells and fibroblasts, indeed, it's still enriched in T cells, but now it's no longer the TPH cells. Now it's the cytolytic CD4 and CD8 T cells that predominate. So you can see that we can predict which particular cell types may be interacting in this context. And so if we take this TB CTAP as an example, and we say, okay, we know what T cells are involved, what are the B cells involved, we can generate a heat map of this type asking for the abundance of different B cell clusters with different T cell clusters, and just highlighting this one. If we ask which B cells associate with the T peripheral helper cells, it's those ABC B cells I mentioned a minute ago, the ones that are implicated as pre-plasmablasts that may be differentiating into plasmablasts and then plasma cells. And so these findings, for example, suggest that in that population of RA patients that have the TB CTAP, the interaction is this particular T cell and B cell population implicating extrafollicular B cell differentiation. So in summary, new and evolving concepts about T cells in RA include, is RA a Th1 disease like we thought before? Well, probably not. It really looks like there's a lot of interferon gamma, but it's mostly coming from CD8 cells and NK cells that I didn't show you. We didn't find much evidence of Th17 cells, although there are some markers that can be associated with Th17 cells. They weren't very predominant in our findings. The main T cell finding that we found is this new one, that about a quarter to a third of the CD4 T cells are these T peripheral helper cells that we think are driving B cell differentiation and antibody production, and an important part of the pathology of the disease. And we found there really are also large numbers of CD8 T cells in the synovium, and they're big cytokine producers, the main producers of interferon gamma. And instead of granzyme B, they have granzyme K. And we described, using the single cell data, a new way to stratify patients with RA based on the cell type abundance within the synovial tissue. And this could be of value in predicting treatments and so on, as I'll suggest in this description of the myeloid cells. So now this is the UMAP of all the myeloid cells in rheumatoid arthritis and osteoarthritis from our single cell studies from the AMP consortium. And I'll just point out, we found a number of clusters of dendritic cells. We found a number of tissue-resident macrophages. These are clusters 0, 1, 2, and 5. These all express CD68, and as you can see over here, some of the markers for those clusters, high levels of MER-TK. They look like tissue-resident phagocytic cells that may be important in tissue homeostasis. In some cases can have even anti-inflammatory functions. In addition, we found a number of inflammatory clusters of myeloid cells, 6, 7, and 4. One group shows a strong interferon signature with high levels of STAT1 and CXCL10. One's a big IL-1 beta producer. Another population expresses high levels of osteopontin. These look like blood monocytes that were recruited in. They lack CD68, express CCL2, and look like they come in from the periphery and become highly inflammatory monocytes. Well, now if we go back to which of these clusters, which of these different types of myeloid cells are in the different stratified populations of rheumatoid arthritis patients, the CTAPs here, M, TM, fibroblast population, and so on, and osteoarthritis, here's what we find. In OA, we find mainly these tissue-resident MER-TK positive macrophages that are not particularly inflammatory. In these other two populations of patients where we don't have many lymphocytes, the fibroblast-enriched population or the endothelial fibroblast myeloid cell population of patients, again, like OA, we see mostly these tissue-resident, not so inflammatory macrophages. On the other hand, When we look at the macrophage enriched or the T cell macrophage CTAP, now we find mainly inflammatory monocytes, and some of the tissue macrophages are becoming inflammatory as well. So you could imagine if you use an anti-inflammatory macrophage therapeutic, it might not work very well in the RA patients with these CTAPs, but it might work much better in the RA patients with these inflammatory monocyte CTAPs. So very important implications for therapeutics. Now I want to talk about one last cell population, which are the fibroblasts. Fibroblasts make up the synovial tissue. It's a mesenchymal tissue, so these are sort of the parenchymal cells of the tissue. But they actually also become very important inflammatory cells in RA and in other diseases. And one way to highlight that is to ask, what's the cell that's the main source of IL-6 in the human rheumatoid arthritis synovium? And it's not actually a leukocyte. It's this one cluster, this one population in the single cell RNA sequencing data of fibroblasts. And I'll show you that cluster here in more detail. So here's a t-SNE of the phase 1 single cell analysis of fibroblasts from the synovial biopsies. And we see 4 major fibroblast populations in dark blue, light blue, going to gray. And this one population I've circled here, the F2 population, this is that population that's very striking in the rheumatoid synovium. It's CD90 positive. You remember there's a lining and a sublining. Well, CD90 is the marker for the sublining. So this is a sublining fibroblast population. It's highly inflammatory, as I just showed you. It makes almost all of the IL-6, but it expresses a lot of the other inflammatory factors that are found, cytokines and chemokines. And what's important is this inflammatory sublining fibroblast population is really profoundly expanded in rheumatoid arthritis compared to, for example, osteoarthritis. In OA, this population is 1 to 2% of the fibroblasts, but in RA, this population is over half of all the fibroblasts. So it's a sublining, highly inflammatory population that's highly expanded in rheumatoid arthritis. So we wanted to learn more about what this population is because it seems to be the driving force for fibroblast pathology in rheumatoid arthritis. So we carried out immunohistologic staining of the synovium, as shown here on the left, where we stained the lining with proteoglycan 4, PGR4, and lubricin, which is considered a lining fibroblast marker here in green. And then we stained with this CD90, the gene name is Thi1, for the sublining fibroblasts, which you can see down here in blue. And then the blood vessels, endothelial cells, are stained in red. Well, when we looked at this, we realized this lining, sublining might not be the right way to think about it. What we realized is what we're calling sublining fibroblasts really are located around the blood vessels and really have a connection to blood vessels. And in fact, that's shown here when we segmented, digitized, and carried out a morphometric analysis. We looked at the ratio of CD90, the sublining fibroblast marker, to proteoglycan 4, the lining fibroblast marker, and we saw that that ratio really correlated with how far the fibroblast was from the nearest endothelial cell. And it's not binary. It's not really lining and sublining. It's a gradient in expression of these two markers that really characterizes the fibroblast and suggests that it's linked in some way to the endothelial cells. And so to ask what might be driving this sublining expanded population, we tested fibroblasts cultured in vitro with a variety of activating factors. We tried inflammatory cytokines like interferons and TNF. We tried Wnt ligands, growth factors. But what we found it was that expanded and activated the transcriptional program of this sublining fibroblasts were JAGID1 and delta-like ligand 4, 2 Notch ligands. When we carried out receptor ligand analysis on the single cell RNA sequencing data from the synovium, we found that indeed the main source of those Notch ligands was the endothelial cells, and the main Notch receptors, especially Notch 3, were found on fibroblasts. Notch 1 was found to some degree as well. And so since these fibroblasts that are in the expanded inflammatory population look like they're being driven to differentiate by Notch ligands and Notch receptors, we knocked out Notch 3 or treated mice induced to have inflammatory arthritis with Notch 3 blocking antibodies. And as can be shown here in the red, this really abrogated the whole inflammatory arthritis process. So in summary, stromal fibroblasts are highly inflammatory in RA. They produce most of the IL-6 and a lot of other inflammatory factors. The sublining CD90 fibroblasts are the ones that are dominantly expanded, and it's Notch3 signaling that drives their differentiation. And if you block this, you can abrogate the whole arthritis, inflammation, and pathologic process. So in my last slide, I'd like to summarize now the reconstructing of rheumatoid arthritis using the single cell data we've identified and a lot of other data from colleagues in the literature. So this is a schematic of the synovium. The lining of the synovium is depicted at the top, the sublining at the bottom, and then the hard tissues of the joint, the cartilage and bone here to the left. Well, if we start here in the middle at the inflammatory process, on the adaptive immune side, we've looked at the T cells, and we found that the most strikingly expanded CD4 population are really these T peripheral helper T cells that help B cells differentiate into plasma cells, possibly by going through ABC B cells, this pre-plasmablast state shown in the single cell data. And then when you get plasma cells, one of their main function is to produce antibodies, including autoantibodies. And autoantibodies and other factors activate macrophages. And we described a number of inflammatory macrophages, mostly coming from the infiltrating blood monocytes that upregulate interferon-stimulated genes, IL-1, and other inflammatory factors that macrophages make. There also, however, are tissue-resident macrophages, which sometimes are anti-inflammatory. I haven't talked so much about them, but they're expanded and associated with remission, as shown by Alavernini et al. In addition, we found the sources of interferon gamma come mostly not from CD4 T cells as previously thought, but from granzyme K CD8 T cells and NK cells from data I didn't show you. At the same time these inflammatory processes are going on, fibroblasts are expanding and differentiating, driven by Notch ligands. The sublining fibroblasts expand as well. And when the inflammatory cytokines from the leukocytes, like TNF and IL-1 from the macrophages, interferon gamma from the CD8 T cells, engage these fibroblasts, it activates them to become this highly inflammatory IL-6 producing expanded population, which dominates in rheumatoid arthritis. And these activated fibroblasts, when they're coming from the lining, as shown by Chris Buckley, drive degradation of the cartilage, and their production of IL-6 and rank ligand partners up with the inflammatory cytokines TNF and IL-1 from inflammatory macrophages, and this activates osteoclasts to erode the bone. And so you start with adaptive immunity, which recruits innate immunity, which activates the stromal fibroblasts, which leads to the damage and destruction to the joint as we begin to reconstruct the process of rheumatoid arthritis. Well, I'd like to recognize the very important role played in this work by the Accelerating Medicines Partnership Consortium. This involves a large number of investigators. Here I just list the primary investigators from the main cooperating sites and our funding agencies, including the NIH, the FNIH, our industry partners, and foundations. And then I'd like to recognize just a couple of people from my laboratory who did some of the work presented. Deepak Rao played a key role in the TPH story. Kevin Way, working with Ilya Korsunsky and Shomura Chowdhury's lab, played the key role in the Notch story presented. Chris Buckley, Jenny Marshall, and Andrew Filer helped us with the fibroblast subset work. And Helena Johnson and Fan Zhang, again in Shomura Chowdhury's lab, provided the CD8 story and a lot of the AMP data that I presented. And much of the computational work carried out was by Shomura Chowdhury's group in a wonderful collaboration that we have together. Thank you very much.
Thank you very much, Professor Brenner, for this really exciting and fascinating lecture. Now it's time for questions. I can see there are already 2 questions on the chat. I will start with the question from Andres Serva. Many thanks for a very insightful talk, Professor Brenner. Do you know whether the CTAPs correlate with patient clinics, such as severity of the symptoms, disease progression, or response to the treatment?
Yeah, no, this is a very appropriate question. And we're working on the computational analysis of those questions. This work is not yet published. It's still in process. We do find some correlation. between the CTAPs, including EFM, that's the endothelial fibroblast myeloid population, and the CTAP, mostly fibroblast population. A higher number of patients with those CTAPs were in the TNF inadequate responder category. More work is going into this. Our study was observational, so it wasn't part of a clinical trial. So we're not powered so well to assess the answer to that question adequately. But I hope that by identifying these CTAPs and stratifying patients in this new way, going forward, we'll be able to see such correlations and perhaps predictions of which drug to use in which CTAP. You know, as we treat rheumatoid arthritis now, we start with one drug or another. We have no way to pick which drug the patient might respond to or which to try first. It's really trial and error and sequence. We hope this kind of stratification may provide a better way to pick drugs more selectively and a better way to carry out trials for new drugs based on the CTAP that fits the pathway that the drug or the cell type that the drug is targeting.
Thank you, Professor Brenner. I think this is a very good segue to the second question that we see on the chat. How could your insights lead to improved therapeutics for rheumatoid arthritis, especially because you also look at pathways, right?
Yes. No, this is exactly the idea. So a brief background, the AMP consortium that I mentioned, which has supported this kind of single cell disease deconstruction, the whole genesis of that consortium came from discussions between Francis Collins, the head of the NIH in the US, and industry leaders, the major pharmaceutical leaders. And what pharmaceutical leaders said is that the problem with drug development, major problem from their point of view, is that so many of the drugs tested in clinical trials fail. 2/3 actually fail in phase 1, phase 2, or phase 3 for lack of efficacy, which suggests that we're not picking the best targets for treating the disease. The drug may neutralize the target, but hitting that target doesn't treat the disease. And so Francis Collins started this consortium asking us to take a new approach to identify targets for drug development or to validate targets better for drug development. And so all of these different cell states, the pathways driving the different cell states, the cell states and cell types that are predominant in the different stratified CTAPs of patients now give us a look at a better selected set of targets that make up the pathways and the cell states that are expanded in those patients for drug development. So it both can help us pick a drug we already have based on the pathways and the cell types that are there, and it is revealing now the pathways. I mean, there's reams and reams of data on hundreds of thousands of cells, and out of that you can pick out predominant pathways. One of the things the CNA, the covariating analysis is showing us is in a CTAP, which particular cell types are interacting. Something, we've made the assumption that when you say inflammatory macrophages, RA has inflammatory macrophages. And what I've tried to show you is from this consortium, we're realizing, no, it's really subsets of RA patients that have inflammatory macrophages. Other subsets don't. You want to target T cells in RA. Well, actually, it's certain subsets of RA patients that have high levels of T cells, not others. And some of them have T cells and B cells. Others have mostly T cells and fibroblasts with not many B cells. So you wouldn't pick a B cell directed therapeutic for that population. So this is really a whole nother way using single cell analyses to think about what drug targets we might pick that would be new therapeutic targets and how to select drugs or test drugs in clinical trials based on this single cell disease deconstruction and patient stratification.
Thank you very much for this answer, Professor Brenner. Another good question. As you consult pharma companies, how easily do they pick up and carry out drug development based on discoveries using single-cell technologies?
Well, the single-cell technologies are new, so I think the answer to that will have to be looking forward rather than looking back. So I don't know there is an answer yet. But for example, the consortium I mentioned has had 6 major pharmaceutical companies help support the costs in partnership with NIH and foundations to do this. And we're actually just starting a second generation of this AMP consortium, expanding it beyond RA and SLE, also to look at psoriatic diseases and Sjogren's syndrome. And so I think this is a big investment both by the research community and National Institutes of Health in the US and by international pharmaceutical companies to further deconstruct inflammatory diseases in this way to identify drug targets and stratify patients. And of course, this has been going on in the cancer field even before we started in the inflammation field. Again, looking for fibroblasts in cancer and T cell types in cancer and different cancer phenotypes, of course. So I think single cells is the way forward to really get a good look at the individual cells, which may be interacting, what cell states they're in, what pathologic pathways are activated. This is this old example of the smoothie, the homogenized mix of things together versus looking at the individual components, fruits or cell types, in the smoothie. We've looked so much at whole tissue and whole blood, and now our ability to get tissue biopsies and do single cell analysis is just showing us the pathology in a whole new way. And as I've emphasized, it's unbiased. So it's discovery based. Then you can take what's implicated and then go back for hypothesis-driven research or validation to figure out if the things you find there really hold up in another cohort and in subsequent analyses. But this is really a whole new window into the nature of immunopathology in the cases that I've been describing.
Thanks a lot for this answer. I would have a personal question as well. As part of the implications of those new approaches that open a completely new way to do research and to identify new targets, new biomarkers, how close are we as well to move away from studies done in animal models? And how can we replace those animal models with better models?
Yeah, that's a great question. And there are several parts to the answer. So as I was referring to having so many drugs fail in clinical trials, as a genesis for this consortium, I think it's fair to say that most of those drugs were picked based on mouse studies or other animal studies. And we've realized that, drugs that treat cancer or inflammation in a mouse model, most of the time they don't translate into human. Sometimes they do, and those are the blockbuster drugs that we have. But for every time one does translate from a mouse study to a human, there are 10 or more that don't. And so we've realized that just picking drugs based on what works in animal models, mostly mouse studies, just isn't adequate. And that's part of the drive for doing these human studies. Now, that said, mouse models are incredibly powerful and terribly useful still, because if you find something in the human by disease deconstruction, you want to go back into an animal model and ask, gee, if I perturb it, if I block it, if I knock it out, does that change the pathology? That's still very helpful as an important indicator that it's likely to block the pathway. But you pick the target from the human study, you maybe validate its ability to influence a pathway in the mouse study. And then another level at which this is going to make things better, you can now take each mouse model of lupus nephritis, each mouse model of RA, each mouse model of IBD, and then you can look at the human single cell data and ask which aspects of it does one particular mouse model versus a different mouse example model. And you begin to realize, oh, one is a good model for the T cell biology, but the macrophages are different. Another is a good model of macrophages or innate immunity, but the adaptive immunity is totally different. Or the response of the tissue parenchymal cells and how they are altered by the inflammation might match what's happening in the human in one model and be different in another model. So we're able now to reevaluate mouse models in a more selective way to recognize what that model is good for in analyzing in the parallel human state. And so mouse models remain highly useful, but now we may be able to use them more intelligently for modeling blockade of a pathway or reflection of a particular aspect of the pathology that's in the human disease.
Thanks a lot for this enlightening answer. Another question from the chat. Thank you, Professor Brenner, for your very nice talk. I would like to know what are your thoughts regarding what is driving the notch-free signaling between endothelial and fibroblast cells.
I think it's probably normal biology. It's normal homeostasis. We think that the notch signaling between endothelial cells and, say, pericytes, the fibroblasts that are around the blood vessels, and the sublining fibroblasts, it's a normal pathway. It's part of the differentiation of the tissue of the synovium, which is really a mesenchymal fibroblastic tissue that has these sublining and lining fibroblasts. What we think happens in RA in the pathologic state is this is turned on. This is, overactive. So there's a lot of inflammatory factors coming from leukocytes that activate the endothelial cells. And then we see a lot more notch ligand production occurring. And then the fibroblasts are getting more notch signals. There's increased differentiation. And then there are a lot of growth factors and other things that drive the hyperplasia, the expansion of the fibroblasts. And then they get activated by these inflammatory factors to become more pathological in their behavior, more invasive, and producing themselves more inflammatory factors. So it's not a unique pathologic pathway. It's one that's just overactive, I think, in the pathological state.
Thank you so much. I think we have no more questions on the chat.
Okay.
So I think we're reaching the end of this seminar.
Well, thank you very much. I've enjoyed talking to you and addressing the questions.
Thank you very much, Professor Brenner. It's been really an exciting seminar and a great talk.